okay?
As good as can be expected. What about me?
Well...
Don't answer that. Hi, humans!
Hey. Thanks for tuning your scanners to another transmission of the Galactic Demystifying Science Alliance.
Or, how I like to call it, the Galactic Science Demystifying Science Alliance. We're here to help you fight against the chaos of misunderstanding, since being able to communicate complicated ideas is the best way to prevent everything from falling apart.
Yes, it is. So, for the last few transmissions, we've been learning about contagion. Where are we at with this?
First was the story of how spontaneous generation gave way to a more detailed, nuanced understanding of how infectious diseases spread.
Finally, humans learned that hawking loogies spreads disease.
Wash your hands, you filthy animals! You too, Mickey.
What? I don't need to wash my hands. There's no way we aren't sharing all the same microbes by this point, Quinn. We're stuck in a metal box together. Anyway.
Okay.
So last time we talked about Koch's postulates. Check out that vid.
Honestly, I'm not a fan, Mickey. These may have been reasonable ideas in the 1800s when humans were first identifying bacteria rather than spirits as the causes of infection. But the postulates were just a starting point.
Starting point for a war on microbes.
Oops.
Took some time to stop fighting battles, but looks like you humans have finally started to understand microbes as collaborators, not enemies.
And you've started to realize that something can be even weirder. That many pathogenic organisms can be present without causing disease.
It's almost as if there's something about the human rather than the bacterium that's determining how the disease will go. Especially when you think about how almost a third of people tested carry around Staph aureus, but most of them don't experience harmful overgrowth.
Or if you think about Typhoid Mary, she was an outwardly healthy chef who was infected with Salmonella typhi. She never got the disease herself, she just gave it to more than 50 people over the course of her lifetime.
Or take tuberculosis. There's this asymptomatic phase inherent in the disease.
And then there's something Koch and Henley couldn't even conceive of, the role of viruses in health and disease.
Well, it's hard to blame them. Viruses are so small and hard to spot. Humans looked for decades before discovering them.
But humans did find them eventually, and it looks like they've even started to understand that they're just as important as bacteria. There's a human microbiome, the world of bacteria hiding out within the human body, and now there's also a human virome.
And just like with the microbiome, taking a closer look at the virome led human scientists to realize the ambiguous relationship between the presence of the virus and the disease it caused.
Even for a virus like HIV, which has killed millions of people around the Earth, there's indication that some individuals have an immune system that's capable of meeting the virus and winning.
Whoa. That's not the case for all viruses on Earth, is it?
It's more difficult to answer, mostly because it's hard to look for that kind of thing. Think about it. If someone's an asymptomatic carrier of a virus, it means that they have some level of virions present in the body, but no active disease.
And disease comes mostly from the body reacting to the replication of the virus, right?
Sometimes viruses can cause organ damage, or even cancer, but often disease does come from the body's reaction to an active virus, which is why asymptomatic carriers are hard to identify. The least abundant, slow-replicating viruses are going to be really hard to detect.
Well, that's because the method they use to identify the bacteria and viruses, called genetic sequencing, depends on being able to read each segment of the genetic code multiple times.
Reading each chunk multiple times gives the scientists confidence that they know exactly what's actually there.
But sequencing has a hard time reading the genetic information of virions that aren't abundant.
Which means that the viral genes that qualify someone as being an asymptomatic carrier can get buried in the background noise that comes from the microbiome or the human's own cells.
So there could be a lot more asymptomatic carriers than they realize. And that's only the first half of the already tricky problem. See, virions They're thought to outnumber cells at least 10 to 1, but they're relatively small and efficient, so they have fewer genes. That means there's way less of their genetic material in a given sample.
This low abundance of genetic material is what makes it difficult to read each section of DNA multiple times. It also means that getting enough starting material to do the study in the first place requires treating each sample to get rid of bacterial or human contamination, and only then amplifying the viral genomes.
So, getting rid of all the other cells is the first place where human scientists run into problems. They filter according to size, which only works for those viruses smaller than cells.
And although there's a lot of viruses that are smaller than cells, some of them aren't, and filtering gets rid of those. No matter what.
Yep, and then on top of filtering, there's another hurdle. Amplification.
Tell us about amplification, Micky.
Remember, virions are so tiny that there's hardly any genetic material compared to a cell. So humans have to take those minuscule fragments and multiply them in order to be able to read them. The problem is that humans know of 7 different ways that viruses package their genes.
Wow.
And they need a separate test for each packaging format.
Amplifying and sequencing one of those formats is a huge amount of work. It would take multiple humans multiple years to actually be able to do it. So trying to capture the whole diversity of virions in a sample is just way more work than a single group of Earth researchers is currently capable of. It's actually so complex that we should just spend a whole episode on it next week.
Yeah, let's do that. And if the human engineers are anything like the alpha-flossine ones, they're probably neck deep in data about this viriome and working together to dig their way out of it into the light.
Sort of. There's a lot of other stuff going on down there. It makes it hard for people to focus on these sorts of questions. But when humans are at their best, taking care of one another and standing against corruption, they can make some pretty serious advances.
Even though you humans can only see part of the picture right now, just keep looking. You'll break through the muck soon.
Yep.
All right, Quinn. I gotta go read up on this virome business.
Sounds good, Micky. I gotta go take care of some of my own work. Bye, humans!
See you next time.